characteristics that is applicable to all Staphylococcus species in general. So the name or the genus name for Staphylococci was derived from two Greek words, staphule, which means bunches of grapes, and kokos, which means berries. And this is because of their microscopic appearance after gram staining. Okay po. So as shown here on the picture to the left, ayan, they are gram-positive, spherical or cocci cells that are in clusters. Now, other general characteristics applicable to all staphylococci are that they are catalase positive, oxidase negative, and they are facultative anaerobes. Okay? So, di ba po, this is something that I've already mentioned no week 2. Facultative anaerobes, they are able to survive in the presence of oxygen thanks to the enzyme catalase. Okay, so catalase decomposes hydrogen peroxide, which can be toxic to bacterial cells. Ayan, so they're also non-motile, and they're non-motile, so wala silang kahit anong form of flagella, and they are non-spore forming. So if ever mapunta sila sa environment with harsh conditions, they won't be able to survive that because they can't produce spores. Now, on blood agar plate, they appear to be small, creamy, or buttery looking, and their colonies may either be white, light gold or yellow, or gray in color. So later on sa mga succeeding slides po, may makikita tayong mga examples ng colony morphology on different types of agar. Another notable characteristic of Staphylococci is that some species can grow in 7.5 hanggang 10% concentration ng sodium chloride. So we could definitely grow them in a high salt concentration media such as the mannitol salt agar, which again is something na elaborate natin later on. Okay, so now let's move along to the specific species. So let's start off with Staphylococcus aureus. This is the most species in the genus, and it can cause an infection when it enters a normally sterile site. Okay, so bale, harmless naman po ang Staph aureus. Considered ito as normal flora. Pero it's only considered as normal flora as long as it stays on the surface of our skin. Okay? Kaso lang po, if they would be dislodged or they would be moved to sites that are normally sterile, that is when they would be able to cause an infection. Okay? So marami po pong ibang virulence factors ng Staph aureus, but its principal virulence factor is the production of the enzyme coagulase. So as I've mentioned earlier, they are halophilic, so they may be grown, or we may be able to isolate them using a mannitol salt agar as shown here on the left. Alright, so isa-isayin na po natin yung mga virulence factors ng Staph aureus. So first is coagulase. So the mode of action of coagulase is by coagulating the fibrinogen in the patient's plasma. So kapag nangyari po yun, the staphylococcal abscesses that were formed during the course of infection, they will form a fibrin layer that can protect them from phagocytosis. Okay? So now, with the fibrin layer, matatakpan po sila. So hindi sila mahanap or hindi sila madedetect ng mga phagocytes. Now, there are two types of coagulase, the bound coagulase and the free coagulase. So the bound or cell-bound coagulase is attached to the cell wall, and it can clot human, rabbit, and even pig plasma by converting fibrinogen into fibrin. Now, free coagulase, it does the same thing, but this can be found extracellularly. Next is staphylokinase or fibrinolysin. So staphylokinase causes a fibrinolytic activity by dissolving fibrin clots, which is the opposite of what coagulase does, di ba po? So kung magpo-form yung mga Staph aureus ng fibrin clot, sila rin po kasi mismo, they can't easily get out of that clot. So they have to, eventually they would have to move from one site to another to spread the infection. So after po nilang maiwasan yung phagocytosis, kailangan din po nilang makaalis dun sa fibrin layer na ginawa nila. That they would do that by dissolving that clot with staphylokinase. So staphylokinase helps Staph aureus to be or to cause disseminated infections instead of just being localized. Next is protein A. Na-discuss na rin po natin to nung week 3, di ba po? It helps avoid phagocytosis by binding to the antibody that should be, or that should supposedly be recognized by the phagocytic cell. Okay, next is hyaluronidase, otherwise known as the spreading factor. So this enhances the invasion and survival of bacteria in tissues by breaking down hyaluronic acid that is present in their connective tissues. Okay, so this would help the bacteria spread further to other sites. Kaya po siya tinawag na spreading factor. Okay, next one is deoxyribonuclease or DNase. So DNase can lower the viscosity of exudates. So by destroying the DNA content of those exudates na napoproduce as the infection progresses. Now, this would give Staph aureus more mobility. Okay? Nakatulad ng hyaluronidase could facilitate the invasiveness of Staph aureus. Kasi po, di ba, wala silang flagella. So they would have to rely on other mechanisms for them to move from one place to another. Next one is lipase, which is a fat splitting enzyme. So this is essential for bacterial survival in places such as the sebaceous glands. So di ba po, na-discuss natin last time when we talked about factors that contribute to the composition of normal flora. Kadalasan po, lipids or fatty acids, they may be toxic to some bacteria, but for the pathogenic strains of Staph aureus, they can produce lipase. So they would be able to survive and colonize areas where humans have sebaceous glands, such as the hair follicles. Now, lipase contributes to the formation of furuncles, carbuncles, and boils. So mamaya po titingnan natin yung clinical manifestations and iisa-isahin natin kung ano yung difference ng mga yan. Okay, so if you've noticed, these 3 virulence factors contribute to the same purpose, which is to help the bacteria spread and infect more sites. Okay, let's now talk about enterotoxins. So enterotoxins A, B, and D, they are responsible for the cases of food poisoning. So enterotoxins are heat stable, or they are effective for up to 100 degrees Celsius even after 30 minutes of heating. So kahit initin nyo po sandali yung pagkain nyo, you may still experience or show symptoms of food poisoning if Staph aureus was the culprit, because the toxin that it produces is heat stable. Naintindihan po ba? So even though this temperature may be enough to kill the bacteria, it's not enough to inactivate the enterotoxin that was produced by the bacteria. So enterotoxins, they're also resistant to gastric enzymes. So yung mga digestive enzymes natin, plus yung hydrochloric acid. And their mode of action is by being a neurotoxin. So they're neurotoxins and they stimulate vomiting. So yun po yung clinical manifestation of food poisoning through the enterotoxin of Staph aureus, no? To stimulate vomiting. Now, the most common enterotoxin is enterotoxin A, while enterotoxin B is associated in cases of pseudomembranous colitis, and it is often found in milk products. So again, the most common one is exotoxin or enterotoxin A. Ayan, next po, here at slide 8, these 2 toxins right here, I've decided to put them together because they're both responsible for causing skin infections. So unahin po muna natin yung Panton-Valentine leukocidin. Panton-Valentine leukocidin, again, this is something that we've already mentioned in passing during week 3, di ba po? It's a cytolytic toxin that can kill macrophages, monocytes, and neutrophils, helping the bacteria avoid phagocytosis. Plus, this can also cause necrosis of the infected skin tissues and also soft tissue infections. So, uunahan na po niya yung phagocyte. So imbis na the phagocyte would kill the bacteria would produce a leukocidin to kill the phagocyte. Next are exfoliatin A and B, or the epidermolytic toxins A and B. So these are proteases that can destroy the intracellular bridges of the epidermis, and they can cause excessive sloughing off of the skin at the epidermal layer, okay? Causing a burn-like effect. So natutuklap po ng sobra at parang nalalapnos po yung balat ng pasyente that are suffering from the scalded skin syndrome because of these epidermolytic toxins. Next is the TSST1, or the toxic shock syndrome toxin. So this is a pyogenic toxin. So it causes almost all cases of menstruation-related toxic shock syndrome. So what happens here is that this toxin stimulates the production of large amounts of cytokines, which would lead to the production of the symptoms. But now, what are cytokines? Cytokines, these are substances that are secreted by certain cells of the immune system that could affect other cells in the human body. So parang sobrang activated po yung immune response ng pasyente that it is now starting to affect the other organ systems, and it's now actually causing more harm than good. Next, virulence factor contributes to antimicrobial resistance. So some strains of Staph aureus can produce beta-lactamase, which accounts for greater than 90% of all the isolates that have shown resistance to penicillin and other beta-lactam drugs. So the mode of action of beta-lactamase is to break down penicillin, as shown here in this reaction. So we start off with penicillin, but after the action of beta-lactamase, it is converted to penicilloic acid. So unfortunately, penicilloic acid no longer has any antimicrobial properties. Okay, now that we know kung ano-ano yung mga virulence factors ng Staph aureus, let's now look into what clinical manifestations or diseases they can cause. So let's start off with cutaneous infections. Okay, so first off is folliculitis. So folliculitis, this is a mild inflammation of the hair follicle or sebaceous gland. So in folliculitis, individually infected po yung bawat hair follicle pasyente. Okay, so there could be many hair follicles that are infected at the same time, as we can see here in this photo, but they are infected separately. Let's compare that with a furuncle. So furuncles are large, superficial abscesses that is an extension of a folliculitis. So dito po, multiple hair follicles or multiple sebaceous glands in one area are infected and they have joined together into this one abscess. And okay po, so as we can see right here, hindi na natin ma-point out individually yung infected hair follicles, unlike here in this first photo. Next are carbuncles. So carbuncles are just multiple furuncles, okay? But now the downside here is that this may now progress to a systemic infection. Systemic infection meaning that the patient may now be experiencing fever and chills. So hindi na po siya simpleng skin infection lang, but now the patient is starting to show other symptoms. So this third photo shows us what a carbuncle looks like. Next is impetigo. This is a superficial cutaneous infection that is common among children, and it is characterized by crusty lesions plus vesicles plus red borders surrounding those lesions. So, ayan, tignan po natin itong fourth picture. So this thing on the patient's top lip, ayan, those are the vesicles. Tapos this one on the patient's nose, that is a crusty lesion. So notice how red its immediate surrounding is. So yan po yung impetigo. Okay. Other infections that may be caused by Staph aureus include food poisoning caused by enterotoxins A, B, and D, as you've mentioned earlier. Staph aureus have also been implicated in cases of acute bacterial enteritis, UTI, septic arthritis, bacteremia, and sepsis. Okay. Let's now proceed to toxin-induced diseases by Staph aureus. So first up is the scalded skin syndrome, as shown here on the photo to the left. So this leads to extensive exfoliative dermatitis caused by the virulence factor exfoliating or yung mga epidermolytic toxins. So this occurs primarily in newborns and in some children. So as you can see here, ayan po, parang nalalapnos o sobrang natutuklap yung balat ng pasyente. Next is the toxic shock syndrome or TSS. So this is a rare but fatal multi-system disease characterized by fever, chills, vomiting, diarrhea, even muscle aches, rashes, and this could rapidly progress to hypotension and shock. Okay, so nabanggit ko na po kanina, di ba? The toxin responsible for this is the pyogenic toxin TSST-1. Okay, let's now look into the microscopic and macroscopic morphology of Staph aureus. So as mentioned earlier sa general characteristics nila, they are gram-positive cocci and they may appear singly, so may paisa-isang cell lang, or they may be in clusters. Now, on culture media, such as the blood agar plate or sheep blood agar, they produce colonies that are yellow, round, smooth, and beta hemolytic, as shown here in this second photo. So notice the cleared out zones of beta hemolysis in this close-up shot. On mannitol salt agar or MSA, Staph aureus will produce yellow colonies surrounded by yellow halos or just in general, a yellow discoloration of the culture media. And this is due to the fermentation of mannitol. So originally po, kulay red yung plate, di ba po? But because this contains phenol red, which acts as a pH indicator, if the bacteria was able to ferment mannitol, this would create an acidic environment. Now, that acidic environment would cause a change in the phenol red from its original color, which is red, tas magiging siya yellow. Next is the chrome agar or the chromogenic agar, like this one in the fourth picture, that is used to identify or differentiate if an isolate is an MRSA. So pag sinabi pong MRSA, it means methicillin-resistant Staphylococcus aureus. So based on the manufacturer, this chromogenic agars may have different indicators. And depending on the color of the colonies, dun po natin malalaman kung MRSA ba yung isolate or not. So usually, if you're using the actual chrome agar, yung mismong brand na chrome agar, yung colonies nila, just like in this photo, they would appear mauve or pink. Tapos, that most likely means that the isolate is methicillin resistant. On to other methods of laboratory diagnosis, we have catalase and coagulase. So catalase, as previously discussed, catalase is an enzyme that helps the bacteria survive from exposure to hydrogen peroxide. Okay, so it converts hydrogen to just, or it converts hydrogen peroxide to just water and oxygen, as we can see here in this equation. Okay, now the catalase test can be used to differentiate catalase producers from non-catalase producers. So basically, staphylococci are catalase positive, and an example of a catalase negative organism would be streptococci. Now, ang reagent po natin dito would be 3% hydrogen peroxide. So using the 3% hydrogen peroxide, the catalase test could be done in a few different ways. So first is on a slide. So just place a drop of Just place a drop of 3% H2O2 on a clean glass slide, and then take an isolated colony from the culture media, and then mix it with the drop of H2O2. So a positive reaction is indicated by bubble formation, as we can see right here. Okay. Ayan po. This may also be done in a tube kung hindi naman po nagtitipid yung laboratory niya sa reagents. Just place 1 ml of 3% H2O2 on a clean glass tube, pick up colonies, mix them, and observe for bubble formation, just like this one. Another way of doing catalase is just by directly dropping the reagent onto the colony on the agar. Okay, so just like in this photo. Ayan, so pagdating nyo po sa internship nyo or sa work nyo, make sure to check with your laboratory staff or section head kung ano po yung method na sinusundan nila to perform the catalase test before you actually do anything. Baka mamaya po ginawa nyo yung direct, pero ang protocol pala ng laboratory is slide method. Next is the coagulase test. So the reagent for this is rapid plasma. So for the slide method, the steps are the same as catalase, but with a different reagent. So place 1 drop of the reagent on a clean glass slide, pick up colonies, mix them together. So a positive result on the coagulase test is indicated by a clot formation, as we can see here in this picture. Dito po sa negative result, kung may makikita man po kayo dyan na mga granules, that's probably just clumps of bacterial cells from the colony that you've been, or that you just picked up. Okay? So kailangan po very apparent yung clotting tulad nito for us to say that it is actually coagulase positive. Next, for the tube method of coagulase testing, again, same steps, different reagent, and the positive result is indicated by a clot formation and cloudiness on the tube, as you can see here in this bottom right photo. All right, let's now proceed to the differential test for identifying Staphylococcus aureus. So kanina po, di ba, nabanggitan natin to, mannitol fermentation test with the or by plating it on a mannitol salt agar. So this is done to differentiate pathogenic strains from non-pathogenic strains of Staphylococci. So pathogenic Staph aureus will produce a positive result with yellow colonies that are surrounded by yellow halos, or just a yellow discoloration of the culture media. So this means that they were able to survive the high salt concentration in the agar, and also they were able to utilize mannitol in the agar as a carbohydrate source. Okay, so as mentioned earlier, MSA uses phenol red as its pH indicator. If the bacteria was able to produce an acidic environment after mannitol fermentation, the color changes from red to yellow. Okay po? So again, MSA is used to differentiate pathogenic strains from non-pathogenic strains. Next is the deoxyribonuclease test, or DNase test. So this is used to check if the pathogenic isolate can produce the enzyme deoxyribonuclease. So marami po kasing strains of Staphylococcus aureus that are pathogenic, but not all of them can produce DNase, which is why we do this test. So the culture medium used here is the methyl green agar. In some companies or in some laboratories, ang ginagamit po nilang stain is toluene blue or toluidine blue. Ayan. So anyway, in this given example, they're using methyl green. Ayan. So here are its optimum incubation conditions. The isolate must be kept at 35 to 37 degrees Celsius and should be stored for up to 18 hanggang 24 hours. So a positive result is indicated by a formation of clear zone around the bacterial growth. So as we can see here in this picture to the left, they tested 3 different bacteria. So they got Staph aureus, Serratia marcescens, and an Enterobacter species. So positive results were produced by both Staph aureus and serratia. So kita niyo po, di ba? Nawala yung color sa paligid nila. Okay, for Enterobacter, it tested negative because although it was able to grow, it did not clear the methyl green on its immediate surrounding. Now, our next question would be, what is the principle behind this test? Okay, so how does it work? So, ang methyl green agar po, or if they use toluidine blue, this would contain DNA molecules. Those DNA molecules are complex or attached to the metal green dye. So if the bacteria was capable of breaking down the DNA molecules, then that also destroys the methyl green and DNA complex. Nagas po ba? So kung na-break down ng bacteria yung DNA content ng agar, madi-destroy din po yung attachment ng methyl green dun sa DNA, which would result in the clearing of, or the clearing out of the area where DNA was able to act upon. So again, that's for the DNA test. Next is the chromogenic test for MRSA using the chrome agar. So MRSA, this is a type of Staph aureus that is resistant to antibiotics such as methicillin or oxacillin. The mode of transmission is through close contact with an infected carrier. So there are 3 types of MRSA. They may be hospital-acquired, community-acquired, or healthcare associated community onset. So methicillin resistance could be acquired after a prolonged stay in a hospital or an extended active antibacterial therapy. And with that, MRSA may also be considered as a nosocomial or hospital-acquired pathogen. Okay. So ayan po, MRSA infections are common among ICU and burn unit patients. Kasi po yun yung mga klase ng patients kung saan nagtatagal po talaga yung confinement nila. Now, for this one, medyo matagal po yung incubation period if we're trying to do a chromogenic test. So we should keep the colonies for 24 to 48 hours before reporting a result. So as shown here in these pictures, depende po sa manufacturer yung color change that would indicate if the isolate is MRSA or not. Okay, so here on figure A, this is an MRSA isolate from one patient plated on a high chrome agar. While this one on figure B, this is also an MRSA from a different patient, but this time it was plated on a chrome agar. Okay, let's now quickly discuss all the other Staphylococcus species that are referred to as CONS or coagulase negative staphylococci. So kung kanina po, we learned that both Staphylococcus aureus is catalase positive and also coagulase positive. CONS are, well, as the name implies, they are catalase positive but negative for coagulase. Okay, so ayan po. First is Staphylococcus epidermidis. This one is considered as a normal flora of the skin. Pero kaya niya pong maging contaminant or kaya niyang i-colonize yung mga medical instruments such as catheters, IV lines, prosthetic heart valves, yung mga ganun po. So, one of the reasons as to why if na-confine po kayo ng medyo matagal, tas nilagyan kayo ng swero, minsan po, di ba, kailangan ilipat yun from one site to another. So, isa po sa mga rationale or isa sa mga reasons as to why they would do that is to avoid colonization of Staphylococcus epidermidis. Kasi po, if that IV line is kept in that same site for too long, ayan po, pwedeng mag-overgrow or pwedeng mag-proliferate yung Staphylococcus epidermidis that is present on the patient's skin. So, ayan po, it may cause infection. Ayan, one way that we could differentiate this from Staph aureus is that it would be negative for growth on MSA. So, hindi po siya makaka-survive sa MSA. Next is Staphylococcus saprophyticus. This is a causative agent of community-acquired UTI, which is common among sexually active females. Now, this adheres most effectively to urogenital epithelial cells. Ang Staphylococcus saprophyticus po, this has been known as the most adherent coagulase-negative staphylococci. So, yung mga mababanggit po natin na cons, siya yung pinaka the best at adhering to surfaces or adhering to its target cells. Now, on the blood agar plate, they are white, opaque, large, and they are non-hemolytic, as we can see here in this bottom right picture. Next is Staphylococcus lugdunensis. This can be identified as a coagulase-negative staphylococci via the tube method. Kasi po sa slide method, it may give up a false positive result or pwedeng maging misleading yung result nito sa slide coagulase. So if we really want to check kung yung isolate po ba is Staph lugdunensis, we should perform a tube method. Last one is Staphylococcus hemolyticus. Ang pinakamahalaga lang po nating kailangan tandaan dito is that it usually has a vancomycin resistance. And so here are the references that were used in the making of this discussion. Thank you so much for listening. Our chat box is now open for questions. So does anybody have any questions po?